So we have covered the cell cycle, the portion called interphase. We said it consists of the G1, the S, and the G2. And now we need to talk about the other part of the cell cycle, that of cell division. So cells go through cell division for some basic reasons. First off, to reproduce. So cells can reproduce themselves by going through cell division. And this may be a means of asexual reproduction, where it's just a single-celled organism and it's going to divide, and now there's two of them. And so that would be considered to be asexual reproduction. Or it could be the production of gametes, such as egg cells or sperm cells, that are required. So cell division is needed to produce them. Or it could be that the organism has lost cells, or cells have been damaged and they need to be replaced, and therefore cell division is required to replace the lost cells. Or it could be that we're looking at a zygote, which is a fertilized egg, and it needs to go through growth stages in order to get bigger. So going from a zygote to an embryo, that will require more cells. So the cell that is present, the fertilized egg cell, needs to go through cell divisions to complete its growth and development. Now in prokaryotes, the process by which they divide is called binary fission. And this is a form of asexual reproduction. So when we use the term asexual reproduction, we're referring to the fact that there is only one parent involved. And that parent is producing cells that look exactly like the original parent. So the DNA that is present is the exact DNA that was in the original cell, or is a copy of it, I should say, that was in the original cell. But there's no differences between the parent and the offspring. So we say they're generating a clone. So the offspring are clones of the parent. Now in prokaryotes, their DNA looks a little bit different. It is a circular strand of DNA, so the ends of the DNA are connected, and they have just one DNA strand. So binary fission is relatively easy. It just refers to the fact that this single circular strand is going to be copied, and those two copies are going to be moved to opposite ends of the cell, and then a new cell wall will form in between those two copies of the circular DNA, and the wall will eventually fuse together to form a solid wall that will separate the cell into two what we call daughter cells. So the offspring from the parent cell are called daughter cells. So we'll make two daughter cells that are genetically identical to the original parent cell. And so this is a form of asexual reproduction because only one parent cell is involved, and we are simply producing two daughter cells that are exactly the same. Now in eukaryotes, the process is a little bit different, and that's because the DNA is different. The DNA is a linear DNA, so it's not circular. Eukaryotes also have a lot more DNA than prokaryotes, so they can have several strands of DNA. And the DNA is also protected within a nucleus. In prokaryotes, they do not have a nucleus because that's a membrane-bound organelle. But in eukaryotes, they do. So to be able to make copies of the DNA and then move those copies about, one of the first things that has to occur is the nuclear envelope, or the membrane around the nucleus, needs to disintegrate. So eukaryotic cell division is a lot more involved than the binary fission of prokaryotes. So let's look at the construction of the DNA and the different forms that it takes while we go through this process of cell division. So we already mentioned that the DNA is going to be copied during the S stage of interphase. And what is copied is a form that's called chromatin. So the DNA exists in the nucleus as chromatin fibers. So chromatin is not just DNA, it is DNA that's wrapped around proteins. So in this image below, it's showing a single chromatin fiber. You have to envision this as being our double-stranded helical DNA strand, but also that there are little proteins kind of along the length of the DNA strand. These are called histone proteins that the DNA is wrapped around. So chromatin is not just DNA, it's DNA that is wrapped around proteins. So during the S stage, that chromatin fiber is replicated. We make a copy of it. So now you have two identical strands of that chromatin. These two identical strands are called sister chromatids. So think of them as being twin sisters. So we have one sister chromatid, and then we have our other sister chromatid. And these sister chromatids are attached at an area along the length of the chromatin fiber that is called the centromere. Now, when you're looking at this image here, so we've talked about the chromatin fiber, we make a copy of the chromatin, and those copies are called sister chromatids. As we go through cell division, this form of DNA is not a really easy form to use. It's kind of like you've taken a ball of yarn and you've unraveled the ball of yarn. And in humans, we have 46, let's say, balls of yarn that have been unraveled. And now we need to divide out those 46 balls of yarn into different cells. That's a tangled mess. All right, so what needs to happen first is we need to wind that ball of yarn back up again. So in other words, we need to wind up the chromatin, and we wind it up into a form that's called a chromosome. So this is what you're looking at here. This says this occurs at the beginning of mitosis. So all that chromatin fiber is kind of wound up on a protein scaffold, and we keep on winding it up. All right. And that is one of the sister chromatids. All right, the other sister chromatid is also wound up. All right, it's just a copy of the other one, so we're going to wind that one up as well. And then where they are attached to each other is our centromere. So what we're looking at are two sister chromatids attached at the centromere. And collectively, the two of them together can be referred to as a replicated chromosome or a duplicated chromosome. So I know all this terminology can get pretty tricky. So chromatin is DNA wrapped around proteins. Sister chromatids are copies of each other. And a replicated chromosome refers to the fact that the chromosome, a single chromosome, has been copied and therefore consists of two sister chromatids attached at a centromere. So we said that cell division can be either mitosis or meiosis, and we're going to start with mitosis. So in mitosis, you're starting out with a single cell, and by the end of mitosis, you're going to end up with two daughter cells that look exactly like the original parent cell. So we're making clones again, in a sense. So this can be a process of asexual reproduction for some eukaryotic organisms, or it can be a means by which we're producing new cells to replace cells that are damaged, or we simply need new cells for growth of the organism. So mitotic cell division can be broken down into phases, just like interphase was. So in this image, it's starting out with interphase in blue, and we're looking at the end of interphase, which is our G2 phase. So we see a discrete nucleus. The nucleus is visible, and the DNA has already been replicated, right? The chromatin has been replicated during the S stage. So there are sister chromatids that are present, but it's all as this loose chromatin fiber during interphase. As we go into mitosis, the very first phase of mitosis is called prophase. And during prophase, we start winding up that chromatin into the chromosomes that you can actually see under a microscope. So the chromosomes become visible. The nuclear envelope also disintegrates, so you don't see the nucleus anymore, and the chromosomes are now kind of free to move about. How they move depends upon the spindle fibers. So these are microtubules that are part of the cytoskeleton that become realigned and reused. So the spindle fibers will reach out from either pole of the cell. So we have kind of a collection on one side here, and we have a collection on the other side, and these spindle fibers reach out from either side of the pole, and they're going to make contact with each of these replicated chromosomes. So hopefully each chromosome, each replicated chromosome, will have a spindle fiber from each pole attached to it. If it doesn't, this is where our checkpoints should hopefully catch that before it proceeds to the next phase. So as these spindle fibers are making connections with our sister chromatids on either side of our replicated chromosome, they can pull the chromosome one side or the other side. And by the time we get into metaphase, these chromosomes, the spindle fiber, should be done pulling the chromosomes one way or the other, and all of the chromosomes should be lined up along an imaginary line in the middle of the cell. So this is what we call metaphase. Metaphase means everything's in the middle. So think M for middle. All your chromosomes are lined up in the middle of the cell. Metaphase is followed by anaphase. And anaphase is a separation phase. So the spindle fibers are now going to contract. And as they contract, they're going to pull on the sister chromatids. So the sister chromatids are going to be pulled towards each pole of the cell. So remember, here's our first pole, here's our other pole of the cell. And as they're pulled, because they're attached near that centromere, the legs or the arms of the chromosomes kind of go flailing backwards as they're pulled towards either side. So anaphase is the separation of sister chromatids. This is followed by telophase. So by the end of telophase, the chromosomes have reached opposite sides of the cell, and now a nuclear envelope or a membrane is going to reform around each one of these sets of chromosomes, right? We just pulled copies apart, and now we can package up those copies into their own nucleus. And each nuclei will have basically the same information, the same types of chromosomes. So we see two nuclei forming here in telophase. Telophase, during telophase, cytokinesis usually starts. And cytokinesis is division of the cytoplasm. So we divided up the nucleus during mitosis. Now the rest of the cell needs to be divided up. So the cytoplasm and the rest of the organelles need to be divided out between the two new daughter cells that are forming. So you'll see kind of this pinching inward of the plasma membrane here, where it's starting to form inward, and that's where our cells are starting to divide. And eventually these two sides will fuse, and we'll get two new daughter cells that are produced by the end of cytokinesis. Now keep in mind, these two daughter cells are going to be exactly the same as each other. We don't have any differences between the two of them.